So, hello everybody. I'd like to welcome you to our second InflammaZoom seminar, today coming from Australia. And I'm really delighted to say I've got two great speakers today. We've got Kate Schroeder from UQ in Brisbane and Kate Lawlor coming to us from Monash in Melbourne. And so our first speaker today is going to be Kate Schroeder from Brisbane. So Kate, do you want to take it away?
Right, let's just make sure the tech's working fine first, as everyone can see and hear.
All good.
Excellent. Right, okay, fantastic. Thanks so much to Claire and the rest of the UK gang for inviting me to speak at this InflammaZoom seminar. It's great. I'm so excited about the seminar series. It's I've been a bit starved for Inflammasome news here in Australia with COVID lockdown, just like everybody else. So it's a really fantastic opportunity to chat to everyone about what we're doing. So I'm going to talk today about mechanisms of inflammasome-driven IL-1 release and also inflammasome inhibition by MCC950. And just before I forget to mention it, we're currently recruiting PhD students for either this year or next year. So please do get in touch with me if any of what I'm talking about today blows your hair back and you're interested in coming and doing a PhD down under. So at the moment, our PhD recruitment is just for students who are within Australia for this year, because Australia will probably have borders closed until the end of the year, but then we'll be opening again for internationals next year. So my lab seeks to understand several aspects of inflammasome signaling. Firstly, how inflammasomes signal, what's the mechanisms of signal transduction, how these pathways drive antimicrobial defense, and also how these pathways drive diseases. And we'll see a little bit later about all of the myriad of diseases that are driven by inflammasomes. We're also very interested in developing and understanding inflammasome inhibitors as disease therapies. And I'll be talking about two vignettes today that highlight signaling and inhibitors, just to sort of give you a bit of a feels of the sort of work we've been doing of late. So I'm not going to belabor the introduction because I'm assuming everybody who's tuning into an InflammaZoom seminar series is going to be a bit of an inflammasome aficionado. So I'll only go through background briefly, but there are many different kinds of inflammasomes that are nucleated by different sensor proteins. For example, all of these NLRs here, and each is activated by very specific types of signals that range from infection through to things like cell stress. And today I'll be mainly talking about the Nlrp3 inflammasome, shown here in the middle, which I always think of as a sensor for cellular unhappiness, because pretty much anything that would make a cell unhappy seems to trigger the Nlrp3 inflammasome. So things like membrane damage leading to ionic flux, or mitochondrial damage leading to reactive oxygen species production. So just again, this is, I think, pretty much the only introduction I'm giving to the inflammasome pathway. So if you're a bit of a newbie, please do listen. Here we have the Nlrp3 inflammasome pathway, which is triggered by endogenous stages signals, things like cholesterol crystals or amyloid beta, as well as by signs of infection. In many of the slides I'm showing today, we often use LPS priming plus Legionella as a stimulus to both firstly upregulate Nlrp3 with LPS, and then Legionella triggers Nlrp3 activation and inflammasome formation, as you can see here. So when the inflammasome forms, you can see there's this nice big oligomer of Nlrp3, which forms a sort of a wheel-shaped structure. And then the adapter ASC binds to this wheel and polymerizes into fibrils, and then caspase-1 monomers bind to this fibril, and this allows for caspase-1 to dimerize. This allows interaction between the catalytic motifs of the caspase monomers to form the dimeric protease structure that is active. So when caspase-1 dimerizes, it triggers self-cleavage in the interdomain linker between the two catalytic subunits, and we get full caspase-1 activation. And Dave Boucher showed a couple of years ago that this active species of caspase-1 is a p33-p10 species that's still bound to the inflammasome. So when caspase-1 becomes active, it can cleave its substrates that include things like gasdermin D, which is a cell executioner protein that forms pores in the plasma membrane, leading to a form of cell death called pyroptosis, which is an inflammatory form of cell death. Caspase-1 also cleaves these unusual cytokine precursors, pro-IL-1 beta and pro-IL-18. These cytokines are then cleaved to their mature forms that are bioactive and go on to trigger inflammatory responses. Now, like all innate immune signaling pathways, the aim of this pathway is really to detect a perturbation in cell physiology and then trigger mechanisms to repair the damage or restore homeostasis. And in many cases, that's what happens. But in some cases, this signaling through this pathway can lead to disease. And as you'll see in a moment, there are a myriad of diseases that signal via this pathway. So I'm going to talk about two stories today, and they are about a long-standing interest of mine in how IL-1 exits the cell. This has been a really intriguing puzzle for decades, since Anna Rubatelli first described IL-1 secretion as being unconventional, meaning it doesn't go through the ER-Golgi route that most cytokines pass through. The real mystery here is that IL-1 is cleaved in the cytosol by caspase-1 and somehow manages to get out of the cell, where it can then trigger biological programs such as fever and neutrophil recruitment, etc., etc. So that will be my first vignette today. And secondly, I'll talk about how we can inhibit Nlrp3 for the treatment of inflammatory diseases. So how does IL-1 exit the cell? This is something we worked on for very many years, and it was a very productive project because it not only produced a paper, which was great, but more excitingly, it produced 3 children. So the 2 postdocs who were leading the study had 3 children between them during the course of this project, which was very exciting. When we started this project, many different pathways had been proposed for IL-1 secretion over the 25 odd years since IL-1 was first described to be secreted by an unusual route. And these include things like secretory lysosomes, microvesicle shedding, exosome release from multivesicular bodies, secretory autophagy, passive release during cell lysis, or somehow that IL-1 translocates the plasma membrane either directly or through a translocation protein or a pore. So basically, every possible way that something could get out of the cell had been proposed at some stage or another for interleukin-1 beta. So it's a very complicated literature. And we reviewed this in 2015, probably at the peak of its complications. And then soon after, a couple of beautiful papers from the Dixit and Shao labs really gave us new insight into this pathway. And so these are the papers published back-to-back in Nature in 2015 that identified gasdermin D as the cell executioner that mediates pyroptotic cell death. Until then, we hadn't known what the cell executioner was. And I'm showing you this figure from Kayagaki and Dixit's paper because it illustrates really nicely something that I'm going to show again. And that is that when you treat cells with a canonical inflammasome stimulus, such as ATP, you see that early cell death is inhibited, but later cell death is it's ameliorated, but not completely ablated by gasdermin D knockout, shown here in red. And actually, IL-1 levels look very similar to LDH release in that early IL-1 release is abrogated in gasdermin D knockouts, whereas later IL-1 release seems to occur independently of gasdermin D. So there was further light shone on this pathway in 2018 when a number of papers, including these papers from the Kagan and Bros labs, showed that IL-1 can exit the cell through gasdermin D pores. So IL-1 can actually pass through the gasdermin D pore prior to cell lysis and that is an exit mechanism for this cytokine. And I guess probably one thing that I should clarify here is that pyroptotic cell death involves many different stages, and so we need to be a bit specific about the way we talk about these stages in terms of the terminology. Gasdermin D pores are mediating membrane permeabilization, but eventually these pores need to accrue and lead to swelling of the cell before the cell will rupture, and then you'll get indiscriminate release of the cell contents. And that's sort of what I would call cell rupture or cell lysis, or cell death, in fact. So Gasdermin D pores do not necessarily equal cell death, and that's nicely illustrated in my next slide, actually. So we were very interested in the data coming out about Gasdermin D because we had, for a long time, been very interested in the response of neutrophils, because we found in this paper published back in 2014 by the amazing Kai-Wen Chen, who I think is tuning in, that IL-1 release from neutrophils is uncoupled from cell death in these cells. So if we treat macrophages or neutrophils with Salmonella typhimurium, here at different MOI, these cells, this is bone marrow macrophages, produce IL-1, and in macrophages this is concomitant with cell death that you can see here, no cell death in the fully inflammasome deficient caspase-1, caspase-11 double knockout. But in neutrophils we get IL-1 secretion that's uncoupled from pyroptotic cell death. There's no inflammasome mediated cell death in these cells under these conditions. So here we've described one of the first situations in which IL-1 secretion can be completely uncoupled from cell death. It was previously known that if you treat, for example, with an osmoprotectant like glycine, you can prevent cell rupture or slow down cell rupture, and that did not slow down IL-1 release. So that was a good indication that membrane rupture was not absolutely required for IL-1 release. But I think this was really compelling evidence that in some situations IL-1 can be released by living cells. And that's a conclusion that other people have also come to since then. So just to sort of continue on this theme, we were interested in whether the IL-1 secretion that we see from living neutrophils is actually secreted via Gasdermin D pores, because I mentioned earlier that Gasdermin D is the cell executioner and also can mediate the passage of IL-1 through the membrane. So I'll show you two data sets here with the flagellum on the left or Salmonella on the right, but basically they do the same thing. They both activate Nlrp3, canonical caspase-1 activation. And in both cases, you see that in neutrophils, early challenge, so at 1 hour with flagellum or 1 hour with Salmonella, gives either fully or partially Gasdermin D dependent IL-1 secretion, but later time points, such as 3 hours with flagellum or 3 hours with Salmonella, starting to give you Gasdermin D independent IL-1 secretion, and this is occurring in the absence of pyroptotic cell death. So there's no Gasdermin D dependent cell death in these cells under these conditions. It should be noted that Gasdermin D knockouts treated with canonical activators can undergo apoptotic cell death, and I'll come back to that. But you'll see in a moment that we can see this in other situations where we're not inducing any kind of cell death, pyroptosis or apoptosis or anything else. So this showed us that IL-1 beta secretion can be uncoupled from Gasdermin D dependent neutrophil lysis, and that Gasdermin D contributes to the early wave of IL-1 secretion, but not the later wave. And we see also in vivo that inflammasome driven IL-1 release can occur independently of Gasdermin D. Here we have mice that have been injected IP with Salmonella typhimurium for 6 hours, and then we collect the peritoneal exudate and look for IL-1 secretion. And you can see here that wild type challenged animals produce nice levels of IL-1 in the peritoneal lavage fluid, but this is completely caspase-1 dependent, so it's inflammasome dependent IL-1, but independent of Gasdermin D. So this Gasdermin D independent mechanism of IL-1 secretion is physiologically relevant in, for example, infection. So this led us to a very interesting question, I think, and that was, what is the precise role for caspase-1? Much of the literature prior to our study had assumed that IL-1 secretion was temporally coupled with cell death, because caspase-1, when it becomes activated, it simultaneously cleaves IL-1 and also activates cell executioner, which we later found to be Gaz-Dermin-D. And so these processes are running in parallel, and Gaz-Dermin-D is then allowing for the release of IL-1. And in earlier papers, before we knew that much about execution, there was the suggestion that caspase-1 was activating some kind of unconventional secretion pathway. So we wondered whether that was really the case, and whether an alternative hypothesis could be true. And our alternative hypothesis was that it wasn't just that caspase-1 was engaging this unconventional secretion pathway at the same time as IL-1 maturation, but indeed IL-1 maturation was required for its secretion through the unconventional pathway. So we were really asking whether IL-1 maturation by caspase-1 was necessary for its unconventional secretion, and indeed whether it could even be sufficient for its unconventional secretion. So that was our, these were our major questions. So we took a somewhat simple approach to this. First of all, we mutated the caspase-1 cleavage site. So there are two caspase-1 cleavage sites within IL-1, this D26 and D117. It's D117 that produces the mature IL-1 P17 cytokine, but there's another site in the pro piece. So we just expressed each of these pro IL-1 constructs, the wild type or the D117A mutant, and we expressed them ectopically with a B5 tag in primary macrophages, and then monitored its secretion. So here we have cells that have been stimulated with LPS plus Negerisin to activate Nlrp3, and we've also treated them with glycine to block non-specific release of cytoplasmic content. So glycine slows down cell rupture, and at this point, which is I think 45 minutes after Negerisin, we have very little indiscriminate release of cellular material such as GABDH. So at this time point, we see nice IL-1 cleavage to P17, and also secretion of the mature cytokine out of the cell into the supernatant fraction. If we mutate D117, we now see, of course, we lose the P17. We only get the D26 being cleaved to produce this P28. And when we treat with glycine, we see very little secretion of P28 and no secretion of the pro form. So this suggests to us that maturation of IL-1 is required for its unconventional secretion, and actually the more of the pro piece that you cut off, the more efficient IL-1 secretion is. So caspase-1 cleavage of IL-1 is required for its regulated secretion. So we then again took a rather simplistic approach and wondered, well, if IL-1 maturation is required for its secretion, is it sufficient? So we then expressed mature IL-1 or pro IL-1 with a B5 tag, again in primary macrophages, either wild type or caspase-111 double knockout cells. And the pro IL-1 behaves just as it did in the last slide, so I won't go through that. But what was really interesting is when we expressed the mature IL-1, we now see mature IL-1 being secreted from completely resting macrophages. This is unstimulated bone marrow derived macrophages, not stimulated with LPS here in the left, or stimulated with LPS as a priming agent. We now see that this P17 form of IL-1 is released by cells that are not treated with an inflammasome activator. And this is, we can show that this is not inflammasome dependent secretion, because if we use the inflammasome deficient ICE cells, we again don't see any effect on P17 secretion in these cells. So we call this slow secretion, and this slow secretion is independent of caspase-111 and is also independent of Gaz-Dermin-D, as you can see here. So when we treat with an inflammasome activator, such as LPS plus Negerisin, we now see really strong secretion of P17, as well as caspase-1, of course, and this strong secretion of IL-1 is caspase-1 dependent and also Gas-dermin D dependent, as you can see probably better here from the retention of the P17 in the lysate of Gas-dermin D knockouts. So this tells us that IL-1 maturation is both necessary and sufficient for its slow secretion from resting cells, even without inflammasome activation, whilst caspase-1 cleaved Gas-dermin D is required for rapid IL-1 beta secretion and strong IL-1 beta secretion. So we wondered next whether IL-1 secretion, so mature IL-1 secretion, involves membrane permeabilization. And this was because of a very interesting paper from the Poligren lab in 2016, which showed a membrane stabilizing agent called punicolagen blocks inflammasome stimulated IL-1 release. We wondered then, we presumed that it would also block inflammasome stimulated release of our ectopic mature IL-1. But we wondered whether it would also block this slow Gas-dermin D and inflammasome independent secretion of mature IL-1. And indeed it does here. If you can see here in untreated cells here on the left, punicolagen absolutely abrogates IL-1 P17 secretion, both in the inflammasome resting cells and in the inflammasome stimulated cells on the right. I should point out that one of the real strengths of this system, being able to just express ectopic mature IL-1 in the absence of an inflammasome activator, is that we're not triggering inflammasome dependent non-pyroptotic forms of cell death. So I mentioned before that in Gas-dermin D knockouts, you can get inflammasome mediated apoptotic cell death. So you are still getting cell death and blebbing from the membranes. This does not occur in, well, we're not stimulating apoptosis in these completely untreated cells here, the resting macrophages that are just expressing mature IL-1. So similar to our results that I've just shown you, there was a very nice paper from the Bruff lab from BC last year, which also showed similar findings that IL-1 beta secretion can occur without cell lysis or death and is blocked by punicolagen. And they also came to the conclusion that membrane destabilization is required for mature IL-1 release. The question is here in these resting cells, what is destabilizing the membrane? And actually we don't have a good answer to that at the moment. Interestingly, here in the Bruff paper, they also show that IL-18 follows a similar secretory route to IL-1, so probably requires similar sorts of cell biology. So of course, we're very interested in why IL-1, mature IL-1, but not pro-IL-1 was released by the cell. And one of the first things we did was just to look at the localization of these two different cytokine forms. And here you can see mature IL-1 on the top and pro-IL-1 on the bottom, in which we've done EM staining, double staining for PIP2 microdomains, shown with the large gold particles here. So they label the plasma membrane microdomains that are rich in PIP2, and mature or pro-IL-1 in the smaller immunogold. I don't know how well you can see it over Zoom, but with mature IL-1, the mature IL-1 and PIP2 probe beautifully co-localize in plasma membrane ruffles and projections, whilst the pro-IL-1 is kept within, retained within the cell and far away from the PIP2 domains and away from the plasma membrane, which you can see here quantified on the right. For those of you who are more used to looking at IF, you can see this here as well. If we express mature IL-1 and look with the V5 tag, we see very nice staining of plasma membrane, as you can see around the circumference of the cell. And this nicely co-localizes with phalloidin, these F-actin-rich projections that macrophages have in their ruffles and their projections. And you can see co-localization here in yellow. So a key question that we had is how maturation of IL-1 actually allows it to traffic to the plasma membrane. Why do these two forms of IL-1 have such different localizations? And here we borrowed from the FGF2 literature, where FGF2 is a very similar cytokine structurally, and actually it's very charged. It's positively charged and has a high PI, just like mature IL-1, which has an isoelectric point of 8.8 and is positively charged in the neutral pH of the cell. What's really interesting is that the pro piece is acidic. So the overall charge of the pro-IL-1 cytokine is actually very negative. It's got a low PI, and so you'd expect this negatively charged pro-IL-1 to repel the negatively charged plasma membrane. So we thought that electrostatic interactions or repulsions may actually drive the localization of mature versus pro-IL-1. And we noted in mature IL-1 this polybasic motif, which was extremely interesting for me because it very much resembles the polybasic motif of FGF2 that's required to interact with PIP2 and is required for unconventional secretion of FGF2. And this is conserved in human and mouse IL-1 beta and in 18. So we postulated that this polybasic motif may allow IL-1 to localize to these PIP2 rich microdomains that we've seen in this previous picture, allowing for their co-localization. So we again did immunofluorescence with mature versus pro-IL-1, and here on the left, mature IL-1, as you can see, beautifully co-localizes, so B5 here and the PIP2 probe here. You can see the co-localization on the bottom in yellow. That mature IL-1 nicely co-localizes in these ruffles of the plasma membrane and also in projections. The pro-IL-1, by contrast, does not co-localize with the PIP2 probe, and you can see very little co-localization here. When we took mature IL-1 and we mutated the polybasic motif, we now see a phenotype that's very similar to pro-IL-1 in that this mature IL-1 can no longer co-localize with the PIP2 membrane domains. So this suggests to us that PIP2 recruits IL-1 to these domains, possibly through the polybasic motif. So we've got this just quantified here. The polybasic motif and pro-IL-1 don't strongly co-localize with PIP2 microdomains, whereas mature IL-1 does. So, so that's the co-localization or the trafficking suggests that mature IL-1 is trafficked to the plasma membrane as opposed to pro-IL-1, which is retained within the cell. But the really important thing, of course, is whether IL-1 release is affected by mutation of the polybasic motif. And indeed it is here. So if we take mature IL-1, wild type versus the polybasic mutant, and let's just look at these columns here, the ones that are LPS plus glycine plus maderosin treated. We can see that inflammasome stimulated IL-1 release is very much diminished in the polybasic mutant, and also our slow Gas-dermin D and caspase-1 independent IL-1 release from resting cells is almost ablated. So this suggests that the polybasic motif is required for regulated secretion of IL-1 through either the Gas-dermin D independent pathway or the Gas-dermin D dependent pathway. So just in summary, here we have inflammasomes activating caspase-1, and this then triggers two different things in the cell. Caspase-1 cleaves IL-1 to its mature form, which is, has a net positive charge. It also triggers the maturation of Gas-dermin D. These are then recruited to PIP2-rich membrane ruffles. I didn't mention this before, but Gas-dermin D is also recruited to these PIP2 positive ruffles and projections of the macrophage. And this then allows, we think, coordinate secretion of IL-1 through Gas-dermin D pores, as well as secretion via these PIP2 domains in a Gas-dermin D independent mechanisms that we still are yet to discover. There was a recent paper from the Plegrin lab also showing IL-1 secretion via a Gas-dermin D independent mechanism, and there they've identified a role for the endosomal protein EEA1. So it could be that these stories are colliding, and we're revealing different aspects of the same gut-determined D-independent secretory pathway, which, as I showed earlier, may require membrane destabilization, and we don't know how EEA1 or other proteins may do that at this stage. So I think I've got about 2 minutes. Is that right, Claire? I've only actually got a couple of slides for the second part.
Yeah, go for it, Kate.
Okay, fantastic. So I just wanted to quickly touch on how some of our work with Nlrp3 inhibitors, because very excitingly, the company that we set up here at UQ to commercialize these inhibitors has now got 2 molecules in that have finished phase 1 and phase 1B clinical trials, and are about to go into phase 2. So Inflammazoom is a CNS active compound that is being tested for fever syndromes and for Parkinson's disease in phase 2, and is a peripherally restricted compound which is also about to enter phase 2. So the time is ripe for inflammasome inhibitors. So I'm going to skip this slide because I'm going to assume that everybody who's interested in inflammasomes is interested because it's so important in many diseases. But if you haven't read Nature Cell or Science for the last 10 years, just take it from me that inflammasomes are important in human diseases. So there are many different ways to target inflammasomes in disease. Currently, anti-IL-1 drugs are in the clinic, but a whole host of other ways to inhibit inflammasomes are now being tested out, and there are startup companies all over the world testing all of these different approaches. So we were particularly interested in Nlrp3 inhibitors, and had the good fortune to be involved with this collaboration with Luca and Neil some years ago, where we described the first potent small molecule inhibitor of Nlrp3, this molecule called MCC950, which has since become a bit of a gold standard for inflammasome inhibition in the field. And I had the great luck to recruit Rebecca Cole, the first author of this paper, to my lab some years ago, where she's been since investigating the mechanism of action of this compound. So as I mentioned, there's intense commercial interest in Nlrp3 inhibitors, including from our startup company, Inflammasome. This slide's probably out of date. There are many more startups now than I've kept track of. But this is a very interesting space for a lot of diseases, including severe COVID, which will be interesting to see what happens with those trials. So we were interested in the mechanism of action of MCC950, and basically we didn't know anything. All we knew that it blocks all forms of Nlrp3 activation, and that it either inhibits Nlrp3 or it's, I've written here in brackets, still somewhat mysterious upstream regulators. You know, we keep thinking we've discovered activation pathways of Nlrp3, and then we keep finding caveats to that. So I guess we'll just keep watching this space. So this is probably the most important slide. This looks very easy, but it was incredibly hard to do. And we only managed it thanks to the chemical wizardry of our colleagues at UQ, Avril Robertson and James Hill, who managed to design an analog of MCC950, which acts as a photoaffinity probe. So you can actually add it to your sample, shine UV light on it, it'll cross-link with its target. And then the cool thing is that on the other end of the molecule, there's a click chemistry handle, which allows you then to biotinylate the probe and then pull it down with streptavidin. And so we did this, and we find that this probe beautifully pulls down Nlrp3 in macrophage lysates, and also pulls down recombinant Nlrp3, showing that it's a direct interaction, or it's likely to be a direct interaction. And importantly, plain old MCC950 that's not a photoaffinity probe nicely competes off the photoaffinity probe, showing that they're both binding to the same epitope of Nlrp3. So to cut a very long story short, we were trying to map the interaction site of MCC950 with Nlrp3, and this slide is really testament to the great veracity of Rebecca Cole in doing her experiments. What she noticed when she was doing her immunoprecipitation of Nlrp3 is that whenever she added 950 to her sample, she couldn't pull down Nlrp3 any longer. And that was a really fascinating finding for us because it suggested that the antibody binds to a similar epitope as MCC950. So we didn't know precisely where the antibody bound, but it was raised against the peptide that was centered on this 306 site, which was extremely interesting for us because it's right next to the Walker B site of Nlrp3. So Nlrp3 has an ATP binding and hydrolysis site, the Walker A and B sites, and this ATP hydrolysis through these sites is absolutely required for Nlrp3's capacity to trigger inflammasome function. So we thought this was very interesting as it might suggest a mechanism in which MCC950 blocks the function of the Walker B site and therefore blocks ATP hydrolysis. And just quickly, we could show that this MCC950 blocks immunoprecipitation of Nlrp3 in all different conditions, whether Nlrp3 was active or inactive. And this was really the most, this is the key message here, is that using surface plasmon resonance, we managed to show that MCC950 very nicely binds to recombinant human Nlrp3. And when you flow MCC950 and ATP in different directions, you can actually see that the addition of both compounds generates a highly stable three-way complex. So this is nanomolar KD, a very strong, tightly bound complex, meaning that Nlrp3 can no longer release ATP or hydrolyze it to release ADP. So when we look at ADP, we see no such strong interaction between the three. And actually, we see ADP does not bind Nlrp3. And this is the really interesting part here, is where if we mutate the Walker B motif, we now still see ATP binding because ATP binds to the Walker A, but we no longer see MCC950 binding. So this suggested to us that we already knew that the Walker B motif was required for ATP hydrolysis and that it was required for Nlrp3 inflammasome function. But what this data suggested is that MCC950 actually interacts in or around the Walker B motif to block ATP hydrolysis and prevent these structural changes that are required for Nlrp3 activation. We published this back-to-back with a really lovely paper from the Legionella lab, which did a lot of structural work using FRET sensors. And they showed the FRET changes that occur during structural rearrangement of Nlrp3 as it goes from being inactive to active. And they also came to the same conclusion that MCC950 blocks the structural changes that are required for inflammasome signaling through this complex. And indeed, they had very interesting data that suggested that you could actually add MCC950 to a preformed Nlrp3 oligomer and dissolve it, making it go back to a monomeric form, which has some interesting implications for ATP hydrolysis in stabilizing the wheel. So very quickly, just to thank all the people who've been involved, past and present. So huge thanks to all the lab, but a huge shout out to Mercedes Monteleone, who carried out the IL-1 project with the assistance of Amanda Stanley, and also Rebecca Cole, who carried out the MCC950 mechanism of action studies and is now lab head at Queen's University Belfast. So thanks also to all of our collaborators who've been wonderful, particularly Jenny Stowe, Matt Sweet's lab, and the labs of Avril Robertson and Mike Jennings. So thanks for your attention. I'm sorry if I've gone over time. Sorry, Kate. I'm happy to take any questions.
Thank you, Kate. That was a great talk. So we've got a lot of really good questions from our participants, so I hope they'll forgive me if I roll some of them up together. A couple of people have asked about, so for example, in a gasdermin D knockout cell that undergoes apoptosis, do you think the IL-1 beta comes out through a different gasdermin, or can an apoptotic cell itself release IL-1 beta? Or do you think there's some chaperone involved? Yep. So for example, Dr. Ling's paper suggesting HSP90 a TMED10 dependent pathway.
Yes, yes. These are all good questions. I don't know if I've got all the answers. What's really, so in gasdermin D knockout bone marrow macrophages in which you're looking at endogenous IL-1, it's very difficult to say how IL-1 is getting out of the cell in the gasdermin D knockout because the cells are undergoing apoptosis and they're starting to bleb and all that sort of thing. And presumably when you take the supernatants off your cells, it will include apoptotic blebs that'll be dissolved by the lysis buffer that you then load your Westons on. So I can't make too much in the way of comments on that, other than to say that when we image those cells, we see IL-1 nicely lining up on the plasma membrane, and actually it looks like it's enriched in the bleb regions. That's in the supplementary data, I think, of our paper. What is clearer is what's happening in bone marrow macrophages when we ectopically express mature IL-1, because then we can start getting Gasdermin D independent secretion of IL-1 in the absence of a macrophage stimulus. So the cells are not undergoing stimulated apoptosis or pyroptosis or any other kind of osis other than their usual turnover. And in that situation, we've previously quantified the amount of IL-1 that's in cells and the amount of just basal apoptosis that's going on in those cells. And I don't think apoptosis could account for the pretty strong IL-1 release that we get. So, you know, the short answer is that we don't know. We also tried those same experiments that were published in the Cell paper using DHFR and to see if IL-1 had to be unfolded and chaperoned through a transporter in order to be secreted. We saw that we did not see any evidence for chaperone-mediated secretion. We found that the cells didn't care whether or not we added the DHFR metabolite, which should stop unfolding and chaperone-mediated secretion. So I don't know. There are cases though in which that could occur, and autosecretion is definitely one of them. There's nice data from the Schenklin lab, which showed under situations of serum starvation that you can get IL-1 secretion through these, autophagy-mediated secretion pathways. In our hands and in the original paper that reported autophagy-mediated secretion from Voyo, in both of those situations, we've found that in serum replete cells or in Optimem, autophagy doesn't have a role to play. ATG5 knockouts, ATG7 knockouts, 3MA treated cells all secrete IL-1 just fine. So I think there may be multiple pathways for IL-1 secretion, which may explain why this is such a complicated mess.
To try and work it out, yeah. Another question, so Murray Clark and Daniel Simpson have asked something very similar. So what biological settings do you think could exemplify the two-motor secretion? And then, do you think the two pathways are redundant in terms of their biological impact or regulated by a specific stimulus?
I don't think they're redundant because there'll be cells that express inflammasomes that don't express Gasdermin D. Maybe not the Nlrp3 inflammasome, because Nlrp3 and Gasdermin D seem to parallel each other's expression reasonably well, but other inflammasomes definitely. And we're already seeing situations in which, for example, Gasdermin D knockout in mouse models of the fever syndrome, I think it was Hiru Kenegan's work recently, showed that then you obliterate IL-1 release, which suggests in some settings it's fully Gasdermin D dependent, whereas perhaps in other settings it could be only partially Gasdermin D dependent, or maybe just that early wave is Gasdermin D dependent and later on it becomes Gasdermin D independent. I should note that we don't know whether the Gasdermin D independent secreted IL-1 is biologically active. It's possible they're in microvesicles or exosomes or some kind of membrane that may prevent it from having its function on the receptor, and maybe there's a second level of secretion regulation then as to how the membrane then is destabilized. I don't know. We haven't done these experiments, but that's something I'm very keen to do actually.
Yeah, interesting. And then a final quick question: does MCC 950 from Evangia inhibit ATPase activity of Nlrp3? She has not seen this. She has not seen this to be when tested on recombinant protein.
Sorry, can you say that again?
Yeah, does MCC 950 inhibit ATPase activity of Nlrp3? This person,
yes, that's our conclusion. Yes.
Yeah, that's your conclusion. Yeah. So this person's not seen it when they've been tested on recombinant protein.
Yeah, it's we've tried to do ATPase activity assays on recombinant protein, and we've never managed to get even our positive controls to work. Other people seem to be able to do it, but we've tried several, and by we I mean Rebecca. She's tried several of these activity assays, and we've never managed to do it. The only way we've managed to do it is by SPR, to get even the positive controls to work. It's our conclusion from our data and from the data from the Polygon lab that we publish back to back with that's what M6950 is doing, that it's blocking ATP hydrolysis to ADP or ADP release. Either of those two things, I guess, is formally possible. But it would be nice to have better systems to actually monitor ATPase activity of the recombinant protein. The problem is that it's almost impossible to actually express full-length Nlrp3 that's active. It's horribly, horribly hydrophobic, and you never get much material that's soluble. So yeah, these are really hideous proteins to work with.
No. Perfect. Thank you, Kate. That was a great talk. And please, I apologise to the participants. We need to move on to Kate Lawlor's talk now. So
it's my pleasure. Thanks, everyone.
Thanks, Kate. That's fab. Yeah. So now over to Kate Lawlor, and hopefully we'll have a seamless transition. Kate, can you stop sharing your screen, Kate Schroeder? Great, Kate, we've got you. Thank you very much. So our next speaker is Kate Lawlor, talking to us from the Hudson Institute in Melbourne.
Thank you, Claire, and thanks to the organisers for giving me the opportunity to speak in this amazing InflammaZoom webinar series. It's pretty hard to come up after the true expert Kate Schroeder in this area, but I'll give it my best attempt. Today I'll be giving three separate sort of small stories on mitochondrial apoptosis and its ability to affect pathogen infection as well as inflammasome activation. So most of you, as Kate has alluded to, will be very familiar with pyroptosis driven by inflammasome activation. Our lab is particularly interested in the Nlrp3 inflammasome. And more importantly, we're also interested in different modes of programmed cell death, such as the extrinsic pathways of apoptosis and necroptosis, as well as mitochondrial apoptosis. And just to introduce these pathways, as they'll come up quite a bit throughout the talk, typically when we study TLR ligation by LPS, in the case of TLR4, or TNF ligation of TNF receptor 1, we study this typically with SMAP-emetic treatment, where we remove the IAP proteins that are core repressors of repoplosome activation. And this repoplosome comprises RIPK3, RIPK1, FAD, and caspase 8. And activation of caspase 8 precipitates a caspase cascade and cellular demolition. And this is typically thought of as immunologically silent in contrast to pyroptosis. For example, when caspase 8 is inhibited, or its activity is inhibited by a chemical inhibitor such as QVD, we then see the formation of a necrosome when IAPs are absent. And in this case, the kinase activities of RIPK1 and RIPK3 are essential for activation of downstream signaling, where RIPK3 phosphorylates the pseudokinase MLKL, and this phosphorylation event allows MLKL to translocate to the plasma membrane and oligomerize, similar to a pyroptotic pool formed by Gasdermin D to induce cell lysis and the release of inflammatory dams. In contrast, when we look at mitochondrial apoptosis, this is really regulated downstream of cell stress responses such as UV damage or growth factor withdrawal. And in this case, it's the fine balance between the pro-survival BCL2 family members and the BH3 only proteins that just determines whether activation of the essential executioners BAX and BAC proceeds. And this activation leads to mitochondrial outer membrane permeabilization, cytochrome C release, formation of a multimeric apoptosome comprising APAF1 and caspase 9. And caspase 9 can then signal activation of caspases 3 and 7 to again induce a silent form of cell death via the release of apoptotic bodies and clearance by professional phagocytic cells. So my lab has been interested in how these modes of cell death might coordinate inflammation and disease, and a number of studies by ourselves and other groups have really shown that when there's a pathogen recognition or a TNF response, the apoptosomes are highly capable in myeloid cells of activating caspase-8 mediated potassium efflux, or in the case of MLKL and necroptosis, potassium efflux via the MLKL pore to allow activation of the Nlrp3 inflammasome. Similarly to reports on caspase-8 activation downstream of other death receptors like FAS, we also know that caspase-8 can cleave pro-IL-1 beta. And it's now becoming apparent that caspase-8 has overlapping functions with caspase-1, where there's been reports of caspase-8 cleavage of Gas-dermin D in bacterial infections. So what has remained sort of elusive in the literature up until now is the role of the mitochondria, specifically back in activating inflammation. And I'll just briefly introduce what is known and go into more detail specifically on inflammasome research later in the talk. So as I've alluded to, back activation can actually lead to an immunologically silent form of cell death, which allows the clearance of damaged, superfluous or infected cells. And in this case, as I said, there's the BH3 only proteins, the activators like BIM, or the sensitizers like BAD that de-repress back activation by blocking interactions with pro-survival proteins, and this leads to this caspase cascade and cell death. But the mitochondria has also been implicated in a variety of functions, including viral RNA recognition by rig-guide MABs that can mediate an inflammatory response for viral clearance. It's a source of reactive oxygen species, which along with a range of DAMPs that are potentially released during mitochondrial dysfunction, like mitochondrial DNA and ATP, have all been implicated in canonical Nlrp3 inflammasome activation. But what about mitochondrial apoptosis? What's very clear is majority of circumstances it appears to be limit inflammation. We know it's critical for cellular removal. It also plays a role in cleavage and activation of IL-33. It was demonstrated back in 2009 by Seamus Martin's group. A number of groups, including the Kyle lab from Monash, have shown that caspase activation, particularly caspase 9, actually limits recognition of mitochondrial DNA and by CGAS to reduce the potential for inflammatory consequences. A variety of studies have also shown that host pathogen interactions, whether it being a direct or indirect, can also potentiate pathogen clearance or dissemination. And more recent studies have actually suggested that even back-to-back mediated autophagy can limit interferon production. But in regards to inflammatory pathways, it's again really showing that caspase-3 can cleave Gas-dermin D to limit pyroptosis, and this potentially is a way that it tries to silence an ongoing inflammatory response. However, on an alternative arm, it's now becoming apparent from work by Steve Tate's group that during mitochondrial outer membrane permeabilization, an IAP degrading factor can be released to stabilize NIC and induce non-canonical NF-kappa B activation of a subset of specific cytokines and factors. And more recently, Feng Xiao and Bishwadix's group characterized Gas-dermin E as another pyroptotic effector that is actually activated by caspase-3 activity, so potentially contributing to inflammatory responses via the release of cellular contents and DAMPs. So there is some literature emerging, but at the moment it's still fairly preliminary in the field, and I'll go back to that a little bit later in the talk. But just to really introduce the pathways and how we sort of come at the role of apoptosis in innate infectious signaling. This work stemmed back from a PhD student in Thomas Nadarel's lab, Mary Spear, and it sort of really started this whole era of studying BCL-XL in the context of inflammation. And so in this study, Mary took advantage of a range of chemical inhibitors that specifically target selective BCL members, including the ABT199 that selectively targets BCL2, ABT737 and ABT263, which target all of the BCL2, XL and W family members, and also 5463 that selectively targets BCL-XL. And she looked at whether these compounds could restrict the replication and survival of Legionella infected macrophages as our gram-negative bacteria of choice for these studies. And so what you can see in untreated macrophages that have received this FLA mutant expressing GFP, the Legionella haemophilus species, you can see that there's unrestrained cell death with an increased bacterial load over time. Our BCL2 targeting drug fails to alter this progression, while surprisingly we found that the BCL-XL targeting drugs ABT737 and 5463 induced some cell death, but this was markedly reduced compared to control, but it did restrict this bacterial replication. And this is perhaps more obvious when you look at these graphs where you can see that ABT199 fails to influence the rate of cell death over time post Legionella infection, while 737 and 5463, you get this initial wave of cell death and then this normalizes and plateaus out. And correlating with this plateau, you can see there's a significant reduction in CFUs in these cultures. And in fact, we see little replication, if any, with 737 or 5463 treatment. When QVD was added, Mary found that this could restore bacterial replication in these cultures. And this replication was, again, occurred when BAX and BAC were removed, which are the ultimate executioners of the drugs, BCL-XL targeting drugs for activation of cell death. So it appears we've got a pathway where targeting BCL-XL using small molecules can block Legionella replication by killing infected macrophages. And we could reproduce this data using BCL-XL knockout macrophages. We were treating them with the FLA mutant and also the DOT mutant to show that the type 4 secretion systems required for this event. And you see that we get around 30% of killing with the FLA mutant, and then this plateau that's characteristic of BCL-XL loss. And again, this is correlated with a severe impact on CFUs in these cultures, showing that BCL-XL deficiency does not allow Legionella replication to occur. So what was causing this protective effect and clearance of Legionella? Well, we went back to think about how these cells may be being killed, where we know that BCL-XL targeting doesn't affect a naive macrophage, and thought about the short-lived protein MCL1, which is rapidly turned over in the proteasome and autophagolysosome. And so here we've again used the FLA mutant and looked over time, and you can see that compared to the other BCL members, we've examined that MCL1, particularly the long form, which is the pro-survival form, is reduced 10 hours post-infection. And so what Thomas and Mary then went on to show using 35S labeling is that when you express a known effector that has been implicated in protein translation inhibition, you can see that protein synthesis is shut down when you express this LGT1 effector from Legionella, and that associated with this LGT1 event is again the loss of MCL1. Importantly, when we combine expression of this 737 with LGT1 effector with 737 and 5463, you can see that we get a heightened level of cleaved caspase 3, and we see an associated increase in cell death with BCL-XL targeting and expression of the LGT1 effector. So what does this mean for in vivo consequences? How does BCL-XL loss affect lethal Legionella infection? In this case, we've used Legionella longbeachii. And so what you can see, we've infected these mice intranasally with Legionella, and then at 8 hours post-infection, looked at the bowel fluid for levels of cytokines. And you can see we completely aggregate inflammasome activation or IL-1 activation, as well as other cytokines, TNF, IL-6. And associated with this is a failure to recruit neutrophils into the lung. And this importantly was correlating with a complete absence of bacterial replication, indicating that this pathway prevents lung inflammation by killing the initially infected macrophages, therefore by preventing damaging inflammation. So just to summarise this section of the talk, we've now shown BCL-XL is clearly an Achilles heel for Legionella clearance. Basically, macrophage survival critically requires two BCL2 proteins, BCL and MCL1. We've also shown that Legionella can inhibit host protein synthesis, particularly of the pro-survival family member MCL1, and this renders macrophages exquisitely sensitive to BCL-XL targeted BH3 mimetics. And so at the end of the study, we kind of proposed that maybe these small molecule BH3 mimetics that were designed for cancer treatment could actually be novel antimicrobials. And this has sort of been followed up in a number of studies, including one with flaviviruses, where BCL-XL targeting did improve pathogen clearance. However, in some cases, such as influenza, it's been detrimental to induce apoptotic killing. And so it is a question that needs to be addressed. Is apoptosis always beneficial for the host, particularly with a variety of pathogens? And one question that was key to my follow-up studies was, can selective mitochondrial apoptosis actually trigger inflammatory responses? And so that brings me to the main part of the talk, which is to look at how back-to-back mediated apoptosis can signal to Nlrp3. So a lot of you will be very familiar with the literature on mitochondria and canonical Nlrp3 inflammasome activation. And after a PubMed search, I saw there's greater than 350 papers that implicate the mitochondria in Nlrp3 activation. And there's over 10 mechanisms that have been implicated in canonical activation, which we recently wrote a review on back in 2019. And including some of these, as Kate alluded to, the production of mitochondrial ROS, but also the fact that there is some suggestion that backs and back downstream of canonical activators can lead to the release of mitochondrial DNA in its oxidized form to activate Nlrp3. And so it's sort of an essential thing to go back and interrogate a lot of these pathways, as a number of them we've actually been unable to reproduce genetically using knockout mice. But there's obviously a lot of crosstalk between cell death and inflammasome pathways, and it's now becoming clear that there's even crosstalk downstream of inflammasome activation, where recently, as recent as about a month ago, caspase-1 was shown to cleave BID to activate apoptotic or secondary necrosis. But one question that still remained unanswered until our recent work was, can specific activation of back-to-back induce inflammasome activation? And to firstly address this, we took macrophages and treated them with a range of protein synthesis inhibitors, cyclohexamide, sylvestrol, and homoharentonin, that will deplete MCL1, which you can see here, in combination with 737 to target BCL-XL. And similar to what we found in our Legionella infected cells, we could see MCL1 loss and even cleavage upon activation of back-to-back mediated cell death. And this was again associated with robust caspase-3 activation. And more importantly, if we incorporated TLR priming to prime the inflammasome, we still got marked levels of apoptosis when BCL-XL was deficient in the cell, and we removed MCL1 using our protein synthesis inhibitors. Critically, when we looked at IL-1 beta secretion, we actually found that loss of BCL-XL and MCL1 activity in the presence of priming was sufficient to activate IL-1 beta, which again pointed to a conserved sort of mechanism where apoptosis might lead to inflammasome activation in certain settings. We then went on to use a recently described MCL1 inhibitor to really pinpoint that it was indeed MCL1 that repressed back-to-back activation in our macrophages. And you can see here with LPS priming S63845, targeting MCL1 in BCL-XL knockouts, was able to induce cell death and also IL-1 beta activation And importantly associated with this IL-1 beta activation was the activation and secretion of the cleaved form of caspase-1, in addition to IL-1, suggesting activation of inflammasome. And just as a side note, to really characterise this pathway, we also demonstrated this was very highly specific to the Bax-Back pathway, as if you remove Bax and Back, you completely abrogate IL-1 beta activity. The next question we had was, well, okay, we know we're getting inflammasome activated. What was the potential sensor that was responding to Bax-Back signalling? And we took a strategic approach and thought about the most likely candidates being AIM2, which is down a DNA sensor and could potentially sense mitochondrial DNA. And again, in regards to canonical or Nlrp3 inflammasome activation, mitochondrial DNA, reactive oxygen species, and a number of other pathways have been linked to Nlrp3 activity. Where we took wild-type Nlrp3 and AIM2 knockout cells, we were firstly concentrated on confirming our SMAC mimetic treatment. So this activates our extrinsic caspase-8 pathway, where we get inflammasome activation and IL-1 secretion, which is Nlrp3 dependent, as you can see here, by the loss of caspase-1 cleavage, but is completely, AIM2 plays no role in this pathway. When we look at our 737 and cyclohexamide stimuli to activate mitochondrial apoptosis, you can see that we get our caspase-1 activation, IL-1 beta secretion as expected. When we look at the Nlrp3 knockout, remarkably, we see this completely blocked, showing that this pathway activates Nlrp3, where responses were normal in the AIM2 knockout. But what became obvious looking at these Westerns is there was a small amount of active IL-1 beta in these cultures, suggesting a bifurcation of the pathway, and that there was another caspase rather than caspase-1 that could actually cleave IL-1 beta. And so we asked the question of how is IL-1 beta activated in the absence of caspase-1 activity? And what we started to notice when we look back at our data is that with ABT737 cyclohexamide, we were seeing sort of around a 50% reduction in IL-1 beta secretion in the Nlrp3 inflammasome deficient cells. And this looked quite reminiscent of what we'd observed previously with our SMAC mimetic to inhibit IAP in these cells. And so we wondered whether in fact we were getting a similar phenomenon to SMAC mimetic treatment, and that Bax-Back activation was actually allowing the release of a mitochondrial SMAC. And this was inhibiting the IAPs and allowing caspase-8 and the repoposome to form to activate IL-1 beta directly, and even potentially activate Nlrp3. And so here you can see just blotting for IAPs fitting nicely with this idea was the fact that we saw a loss of CIP1 and XIAP, which are the core repressors of the repoposome. And this sort of pointed us in the direction that indeed we might be tracking a caspase-8 response. So does Bax-Back signaling lead to caspase-8 mediated IL-1 activation? Well, in the first set of experiments we did looking at the Bax-Back knockouts, it seemed very clear that caspase-8 activation was dependent on Bax-Back being present without apoptotic stimuli, and that this correlated with a complete loss of caspase-1 activation. However, when we looked at bone marrow-derived macrophages from two separate caspase-8 knockout lines, we saw that though there was a block in IL-1 beta secretion, it wasn't 100%, and in fact it seemed to be around the 50% difference we saw in our Nlrp3 knockouts. And then using the comparison with our extrinsic apoptotic stimuli compound A, where we can see IL-1 beta secretion and caspase-1 activation, and this is completely dependent on RIP3 and caspase-8. When we look at ABT737 and cyclohexamide, you can see that this is only partially blunted when you're looking at caspase-1 activity. In fact, there's considerably similar levels of caspase-1 activity, and it's really only the IL-1 beta levels that are blunted. So from this data, it seemed to us that in the Bax-Back activation was actually bifurcating to induce an IAP degrading factor to allow IAP loss to drive caspase-8 activation, And Vaxback could also activate Nlrp3 in a separate separate unknown mechanism. So getting on to how does Vaxback activate Nlrp3, we looked back and thought about interrogating the apoptosome and specifically downstream caspases, caspase 3 and 7. And what we found when we used caspase 3-7 double knockout fetal liver macrophages is that IL-1 beta secretion was completely blocked when we activated apoptosis in this system, as was cell death. And this is quite an impressive reduction, suggesting that caspase 3 and 7 were the caspases upstream of Nlrp3. In an interesting aside, we actually saw that with nigericin stimulation, that caspase 3 and 7 activity was actually required to blunt nigericin and canonical inflammasome activation, potentially suggesting a role, again, as have been alluded to, for caspase 3 in activating Gasdermin D to prevent ongoing pyroptotic inflammation. Looking at our Western blots, this was sort of a summary of some of a detailed analysis, but you can see that again, caspase 1 activation was blunted in our caspase 3 and 7 knockouts. But surprisingly, what we actually found was, in fact, caspase 8 was also blunted downstream in the absence of caspase 3 and 7. And so this was quite a surprising finding, as it now places these terminal effector caspases upstream of the initiated caspase 8 in its stringent cell death, and also positions caspase 3 upstream of the inflammasome. So how does caspase 3 and 7 signal to the inflammasome and cause IL-1 secretion? An obvious candidate was that caspase 3 was cleaving the recently discovered Gasdermin E to allow pore formation and potassium efflux to activate Nlrp3. But what we found was, in fact, that Gasdermin E, and this was work done in Feng Xiao's lab, was not required for apoptotic IL-1 secretion or activation, and that MCC 950 reduced levels in both the Gasdermin E and wild type to a similar level, suggesting it had no role in inflammasome activation. And similarly, we found that Gasdermin D was not essential for IL-1 release upon Vaxback activation. So to summarise a long story, we then went on to look at potassium as a candidate, where we'd shown in 2 other modes of cell death the potassium efflux was essential for Nlrp3 activation. And indeed, when we cultured our cells with high extracellular potassium chloride, you can see with our apoptotic stimuli, we could completely block caspase 1 activation. And so this led us to sort of a summary model, which has now been built on by groups such as the Bros lab, where we now know that when Vaxback is activated in the absence of BCL-XL and MCL1, you get activation of caspase 3 and 7 via apoptotic signaling, and this causes a membrane channel alteration, allowing potassium efflux to activate Nlrp3. And in a separate arm, we now know that we get an IAP degrading factor, de-repressing caspase 8. However, caspase 3 and 7 is still required to activate caspase 8 in this scenario to directly process IL-1. And so it was at this point we thought, well, we've got a really interesting story. However, at the moment we're using anti-cancer drugs. Is there a realm for it to be involved in pathogen responses? And so we moved back to our collaborator, Thomas Nuttall at Monash, and his talented postdoc Pankaj Dio, who was studying gram-negative bacterial derived OMB responses and their ability to cause mitochondrial dysfunction, induce cell death. And so we asked the question, could we actually influence inflammation, specifically inflammasomes? And so this built on work from Pankaj back in 2018, where he analysed these Neisseria OMVs for their composition and found toxin POR-B that could localise the mitochondria, specifically target the mitochondria. And it also led to a rapid cell, or relatively rapid cell death that was associated with caspase 3 activity and membrane permeabilization. And in fact, that the cell death could be blocked with a pan-caspase inhibitor, QBD. So in his next paper, he really wanted to answer the question, what was the specific pathway of cell death, and could these cell death pathways influence inflammasome activation? And so Pankaj firstly asked, using caspase 11 knockouts, he isolated E. coli OMBs to really use a positive control on his system and show that in fact These E. coli Ombs induce rapid cell death, and that this is critically dependent on the caspase 11 inflammasome and pyroptosis. While in comparison, when he used Neisseria Ombs, he could show that there was, in the wild type cells, some cell death. This is slightly delayed in cell death, and that when he used the caspase 1 and 11 knockouts, he could delay cell death for a few hours, but eventually the cells succumb to death, suggesting another pathway other than pyroptosis. So why were we seeing this sort of mainly a blip of pyroptosis, but eventually another mode of cell death, most likely apoptosis? To summarise a lot of work looking at the proteome as well as lipid A composition in the LPS within the OMVs, Pankaj found that there was low abundance of, shown in blue, of phosphate negative charged lipid A compared to this phosphoethanolamine modified lipid A that is less likely to interact or actually block interactions of the phosphate with caspase 11 to activate the non-canonical inflammasome. But what was interesting is when Pankaj looked at inflammasome activation in these cells and measured IL-1 beta, you can see that both the E. coli OMV, as you'd expect, and the Neisseria OMV were critically dependent on caspase 11 and caspase 1 and 11 for IL-1 secretion. Although whether this blip is due to the small amount of pyroptotic cell death that might be observed earlier in these cultures is a little bit unclear, but it's still very interesting to see a divergence of these two pathways. He then went on to characterise the responses of numerous OMVs, including Neisseria, comparing to uropathogenic E. coli and Pseudomonas species, and found that the OMVs were inducing a BAX-BAC dependent form of cell death, or more specifically, and it's a bit hard to see in these graphs, a BAC dependent form of cell death, where you can see that these cells were protected from apoptotic signalling. And remarkably, this was less important in this response was BACs, where we tend to really think of BACs and BAC as a combined functional effector protein to induce ultimate demise of a cell. So how do the OMVs trigger BAC-mediated apoptosis? We thought back to our previous research in Legionella and thought, well, do these OMVs influence protein stability? Could MCL1 loss contribute to sensitized macrophages to death? And what Pankaj and Thomas found was these OMVs do in fact alter protein synthesis, shown by this radio labeling of protein. And this is comparable to, again, cyclohexamide treatment to inhibit protein synthesis. Looking by Western blot, you can see that MCL1 long levels were significantly blunted, but only between the 6 and 24-hour time point. And this short form is more of a killer form, or thought of as more of a killer form in these assays. Importantly, Pankaj would show that the stability of MCL1 would actually dictate responses, where this mouse has a stabilised MCL1 protein, and you can see that this represses the mitochondrial apoptosis we observed. The next question was, well, similar to our in vitro work, could we actually exacerbate OMV-induced apoptosis and inflammasome, potentially activate inflammasomes if we remove BCL-XL and MCL1? And we nicely were able to show that BCL-XL deficiency exaggerated OMV responses with accelerated cell death compared to controls. And we saw a significant increase of IL-1 beta activation. And we found this for a number of different selective gram-negatives. So we sort of at this model now where we know that the OMVs target the mitochondria via a toxin, such as PORB. It can also influence protein stability of MCL1, which unleashes back to cause apoptosis. At the same time, in this slower mode of death, LPS lipid binds caspase 11 less efficiently to activate Gasdermin D and pyroptosis, and at the same time causes potassium efflux to activate the inflammasome. When we have BCL-XL absent, we now know that this really drives a BAC-mediated response, and it accelerates caspase 3/7 activity to apoptosis. And it also potentially can influence activation of the inflammasome, whether this is secretion and or activation is less clear, but it appears that there's likely to be a contribution of our in vitro pathway that we published in last year or the year before. So the final question we had was, is this relevant? Could BCL-xL mimetics actually change inflammatory responses to OMVs in vivo? And what we surprisingly found when we injected these OMVs to induce a sort of septic shock was that caspase 11 was not required for this illness. And in fact, we saw increased IL-1β serum levels in the caspase 11 knockout. I haven't shown it, but we then went on to confirm that BAX and BAC do actually play a role in activation of IL-1β secretion in this OMV-induced IP model. And if you knock out BCL-xL, you can see we exacerbate this response by increased weight loss, as well as an increase in IL-1β serum levels. So in the case of in vivo, it's slightly different to what we found in vitro. It appears that mitochondrial apoptosis, more dominantly triggered probably by the toxin encapsulated, as well as other effectors, activates apoptosis preferentially, and a pathway to caspase 3/7 inflammasome activation may be occurring. And it's less able to utilise the lipid modified that's the modification that's found within the OMVs to activate the non-canonical pathway. So just to finish, and I've probably gone over time, I'd just like to acknowledge my lab at the Hudson, including past members, but really want to point to the people that really drove this work. Mary Spear, who's a talented PhD student who's now my postdoc, and is following up looking at other novel regulators of cell death, pioneered the sort of area on Legionella and BCL-xL when she was in Thomas's lab, and supervised by James, who was my old boss. The second part of the story was led by James and myself, and there was a lot of colleagues, both WeHi at Monash, as well as external collaborators, Seeming Man, Vishva Dixit, that provided mice, and Feng Xiao's group actually performed the Gasderm B experiments. And the third story was really led by Pankaj and Thomas, where James and I sort of made major contributions on the overall cell death inflammasome field. And I'd just like to acknowledge my funding. Thanks.
Thank you, Kate. What a great talk. That was really, really interesting. So first up then, a question from me, which is, it's really interesting, isn't it, the complex interplay of all the cell death pathways. So do you think the ones that predominate depend upon the kinetic response of the cell death pathway? So the ones that kick in first is the ones that kicks off fastest?
Yeah, I do think that's right. And I think that's why Pankaj was seeing, you know, the pyroptotic phase, even though the lipid had modifications that would inhibit caspase 11 binding. There was enough, you know, of the proper form to actually induce probably a rapid pyroptosis that was then just shut down by the mitochondrial apoptosis coming in. And it was only in that phase, which is really slow, when you accelerated it again, did you suddenly see another brake being released. And I think all the pathways are the same. And if you take away one mode of cell death, it's like the cells find another way to die, whether it's a bit slower or in some cases it actually provides more machinery. So if you inhibit the Nlrp3 kinase activity, for example, with apoptotic stimuli, you actually push caspase 8 more strongly to activate IL-1 and cell death. So it's like it's all a platform that moves around. Yeah, no, that makes sense. And then what about gram-positive bacteria? I have done nothing on gram-positives at all. So that's something that would be really interesting to look at. Yeah, I agree. Most of the stuff is focused, isn't it, on gram-negatives. Kate Schroeder says, just an aside, does BAC's mitochondrial signaling influence bacterial killing by mitochondrial ROS? We haven't looked at mitochondrial ROS, and I think that would be interesting to look at, because we really still don't know with the bacterial killing, for example, in the IMVs, we haven't formally proven whether it's caspase 3 and 7 or whether there's an upstream event that might be triggering the inflammasome. We actually know the inflammasome does influence the cell death, so it is blocked with MC16 on 50. So there's, you know, there's 2 steps that could be being interfered with in the, you know, the mitochondrial signaling pathway. So there's still quite a lot to answer.
Yeah, well, tons to answer. Thank you. That's a beautiful talk. So it just remains now then for me to thank our brilliant speakers this morning, Kate Schroeder and Kate Lawlor. It's been really, really interesting science that we've all been lucky enough to listen to this morning. And secondly, to invite you all to join us in a month when I'll be going to Boston, I think, for our next InflammaZoom seminar. So just thank you very much, everyone, for joining us, and see you all soon.